Hello everyone, it's me Ali, a nursing student here in Davao City. So hey guys, so for today's video, we're gonna talk about our new Parasite. So are you ready to know those Parasite? So our new Parasite is called about Entamoeba Histolytica. So we're gonna talk about its distribution, habitat, life cycle, morphology, diagnosis, treatment, and prevention and control. So are you ready to know those?
Oh yes.
Because I am also ready. You're ready, I'm ready, we are ready. Let's go! So our parasite for today is the Entamoeba histolytica. So the disease that we can get on this parasite is what we call amoebiasis. So it is an intestinal or bowel illness caused by a microscopic parasite called Entamoeba histolytica. So these are the consideration to know this parasite through its distribution, habitat, morphology, life cycle, pathogenesis and clinical features, immunity, diagnosis, treatment, prevention and control. So first we're going to talk about its distribution. So the distribution of Entamoeba histolytica is global, with higher prevalence observed in areas with poor sanitary conditions. Places with highest rates of this parasite including India, Africa, Mexico, Central America, and South America. So next we're gonna talk about its habitat. So the habitat of Entamoeba histolytica is in the colon and cecum of the large intestine in the human body. So this parasite is transmitted through the ingestion of contaminated food and water. So now we're gonna talk about its morphology. So the Entamoeba histolytica has a unique morphology that helps distinguish from other amoeba species. So its size ranges from 10 to 60 micrometers in diameter, and it has a characteristic shape that can vary round to oval. So the cytoplasm of Entamoeba histolytica is granular and contains vacuoles. And the Entamoeba histolytica has a single nucleus that is usually centrally located within the cell. So the Entamoeba histolytica can form cysts, which are a dormant and resistant storage of a parasite. So next is the life cycle of Entamoeba histolytica. So in the first stage, cysts are typically found in form stool, whereas trophozoites are typically found in diarrheal stool. Infection with Entamoeba histolytica occurs via ingestion of mature cyst. So in the second stage, from fecally contaminated food, water, or hands, exposure to infectious cysts and trophozoites and fecal matter during sexual contact may also occur. On the third stage is the excistation occurs in the small intestine, and the fourth stage is the trophozoites, which are released, which migrate into large intestine, and also multiplication into trophozoites and cysts. So in the fourth also, which is the trophozoites, may remain confined to the intestinal lumen, with individuals continuing to pass cysts in their stool. So trophozoites can invade the intestinal mucosa or blood vessels, reaching extraintestinal sites such as liver, brain, and lungs. So trophozoites multiply by binary fission and reproduce cysts, and both stages are passed in the feces. And cysts can survive days to weeks in external environment and remain infectious in the environment due to the protection conferred by their walls. Trophozoite passed in the stool are rapidly destroyed once outside the body, and if ingested would not survive exposure to gastric environment. So next, we're going to talk about the pathogenesis and clinical features of Entamoeba histolytica infection, or also known as amoebiasis. Can vary depending on stages of infection and the host immune response. So the first is infection. So Entamoeba histolytica primarily infects the large intestine by ingestion of contaminated food or water containing cysts of the parasite. Next is the intestinal disease. So, in most cases, the infection remains asymptomatic. However, in some individuals, the protozoites can invade the intestinal mucosa, leading to amoebic colitis. So, this can cause symptoms such as abdominal pain, diarrhea, and mucoid stools. The third one is the extraintestinal diseases. So in some cases, the protozoites can penetrate the intestinal wall and spread to the other organs, leading to extraintestinal diseases. So the fourth one is the immune response. So the pathogenicity of Entamoeba histolytica is attributed to the ability of protozoites to produce enzymes and toxins that cause tissue destruction. So, next is the immunity. So, the immune response plays a crucial role in the control and clearance of Entamoeba histolytica infection. So, here are some key aspects of the immune response of E. histolytica. So, first is the innate immune response. Second is adaptive immune response. Third is the cell-mediated immunity. Fourth is the immune evasion mechanisms. So first is the innate immune response. So the innate immune system is the first line of defense against E. histolytica. So it includes physical barrier, for example is the intestinal mucosa, antimicrobial peptides, and cell such as macrophages, neutrophils, and natural killer cells. So the second one is the adaptive immune response. So the adaptive immune response is specific to Entamoeba histolytica and involves the activation of the T cells and the B cells. So the third one is the cell-mediated immunity. So cell-mediated immunity primarily mediated by T cells. So is crucial for controlling Entamoeba histolytica infection. So the last one is the immune evasion mechanisms. So Entamoeba histolytica has developed various mechanisms to evade the host immune response. It can modulate the production of cytokines and interfere with antigen presentation and inhibit the killing mechanism of immune cells. So let's go back to cytokines. So what is cytokines? So, cytokines are crucial in controlling the growth and activity of immune systems and blood cells. So now let's move on to the next, which is the diagnosis. So first is the stool examination. Second is the antigen detection. Third, the serology. So first, let's talk about the stool examination. So it is a fresh stool sample is collected and examined under a microscope, and the stool examination is the most common method for diagnosing Entamoeba histolytica infection by examining stool samples for the presence of cysts or trophozoites. So the second one is the antigen detection. So the antigen detection test can be used to detect specific proteins or antigens of Entamoeba histolytica in stool samples. So these tests are more sensitive and specific than microscopy and can help differentiate between Entamoeba histolytica and other non-pathogenic amoeba. Next is the serology. It is a serological test detect antibodies produced by the host in response to Entamoeba histolytica infection. So the tests are useful for diagnosing invasive amoebiasis and can be used when the stool examination or antigen detection is inconclusive. So we will now proceed to the treatment. So the treatment of Entamoeba histolytica infection typically involves the use of a specific medication to eliminate the parasite. So first is the antiparasitic medication. It is the primary treatment for Entamoeba histolytica infection. It is the use of antiparasitic medication. It is the most common use medication is metronidazole, which is effective against both trophozoites and cyst forms of the parasites. So the next one is the luminal agents. So in addition to antiparasitic medications, luminal agents may be prescribed to eliminate the parasite from the intestines. So these agents such as deloxinide, furoate, or paronemacin are not absorbed in the bloodstream but act locally in the intestines to kill the remaining trophozoites and prevent reinfection. So the third one is the treatment of invasive amoebiasis. So in cases of invasive amoebiasis, where the parasite has spread through other organs, additional intervention may be required. So the last one, which is the supportive care. So the supportive care measures such as maintaining hydration and electrolyte balance may be necessary in the individuals with severe symptoms or complication of Entamoeba histolytica infection. So let's move on to the prevention and control of this parasite. So first is the proper disposal of sewage. Next is handle soiled diapers carefully. Third one is the washing hands with soap and water after use of a toilet. Fourth is drink boiled water and sealed water when traveling. The last one is maintain proper washing and cleaning the food before use. Thank you for watching, everyone. Hope you gain a lot of knowledge on this video. So see you on my next vlog. Bye!